So, this is our presentation over our experiment of collection, culture, and identification of wild ciliates. So, we started out actually collecting samples of local wild ciliates using Ziploc bags and inserted into golf ball retrievers. We went to Fern Hill and collected 4 samples just of the water there, and we made sure to actually get some sediment, which is where the ciliates and their prey would be found. When we got back to the classroom, in 2 out of 4 of the bags, we put traps that were made with ciliate food and antibiotics to stop the growth of competing organisms. And those traps were really, really successful. As we can see from the figure 2, there was quite a large population of ciliates when we examined the traps under the microscope.
To isolate our ciliates, we prepared wet slides from all 4 of our bags, the traps that we had dumped out the morning after, and then our hay infusions that we collected a couple of weeks ago. And we looked at each of those under a microscope, kind of saw which ones had ciliates, if they did have ciliates, if they didn't, and if they did, whether the population was dense or if there was only a few and they weren't really going to be good to work with. And then from there, we prepared media for 2 48 well plates that we put each of our samples into and divided them up so that we could further culture them.
After about a week, we let them culture until the next lab class. After that was done, we extracted DNA from 8 samples in total. We looked at a bunch of the wells under the microscope to observe the populations, their health, how large the populations were, and then to extract the DNA, all we had to do was add samples to the degrading chemical, the Chelex, which breaks down the cells to release the DNA. And then we just incubated and vortexed them to solidify the DNA into one single pellet. You can see in figure 6 that we did end up with good ciliate populations to extract DNA from.
So this is our PCR results, and you can see in sample AT2, AT6, and CT4 that they came back negative. They didn't show up. And that also happened further down in sample AT4. And so that left us with samples CT5, CT6, AT3, and CT1. But the bands in CT6 and AT3 were really smudged, and they didn't come out very clear, so we opted not to use those. And instead we used CT5 and CT1 because they were the clearest and they were really bright. And even though all of our lanes had like primer and dimerization, these 2 had the best bands to work with.
Once we sent our samples for sequencing and the sequences came back, we uploaded them to DNA Subway. And once we cleaned up the sequences and combined the forward and reverse for each sample, we did a blast search for the closest relatives. And the closest relative to our own sample is Tetrahymena nipissingi, which has 49 mismatches out of a base length sequence of 666, which is a 7.36% difference. And if we go by what was recommended in the Kerr paper, a difference of more than 1 or 2% as indicating a separate species from a known sample, then our sample is clearly not this known species. So that was the first indication that we had that our sample was of a new species of Tetrahymena.
This is our sequence we generated it using CLC Viewer, so you can see all the pretty colors. And if you look down at the consensus at the very bottom, and then from there you go up, you'll see our MLC1 and MLC5. Those are our two samples. And if you just kind of look across, you can see that there's noticeable differences, and it doesn't necessarily match the consensus very well. A lot of the bars aren't very high. There wasn't that great of a match. And even compared with other species, you can see there's mismatches all over the place. So going off of like that 2% difference, you can kind of see that we had more than a 2% difference.
Lastly, what we did was to use that information to generate a neighbor joining tree. And we used the information that we got from the BLAST results to compare our samples down here to the closest relatives. And we can see that they are clearly some species of Tetrahymena, as they fall into this general Tetrahymena family. However, they're more closely related to each other than to any other species. They don't fall very closely to any other known species. So that's, again, more evidence of our species being a new species rather than a known species, such as the Tetrahymena rostrata or canadensis. So we have a lot of evidence that there are, in fact, some new novel species here in the Pacific Northwest, and which is big news because no one really knows a lot about the distribution of Tetrahymena. So this is important information for studying this incredibly useful species. And that concludes our presentation. Thank you.